Well, here we are with Module 5. Getting my pen ready here. Cell structure function part 2. And I'm going to warn you right off the bat that this is a much longer module, a lot more lectures this week, even though I just divided the chapter in half. I'm going to go a lot deeper in some areas than the book does. And I will let you know I'm only going to test about the depth of the level of the depth of the book, but I do think some of this information is important, so that's why we are going to dig a little deeper, especially because this is a biology course for future fitness professionals. So let's start looking a little bit more in depth in that cell structure and function, and we're going to look at basically how that DNA makes proteins. So it's all about protein production. And so it all goes back to my little triangle that you're sick of, I'm sure. And over here we have DNA. And we know that it's going to basically encode proteins. So now we're going to really talk about, okay, how does this happen? And this is really a question for molecular biology. And that's where we're going to find out the real answers a little bit later in this quarter. So maybe a better way of phrasing it for this lecture today, instead of how does this happen, maybe where does this happen? And it starts off in that nucleus, goes to the ribosomes, and what we're going to call the endomembrane system. So these are the areas within the cell that have to do with protein production. So starting off with the nucleus, we already know a bit about the nucleus. We know that it houses DNA, right? It houses DNA and associated proteins called chromatin. And it's bound by that porous nuclear envelope, that nuclear membrane. And porous because it has these holes that proteins are going to be going through when they're, ribosomal RNA is going to be going through when it's made. Right? There are those pores. And it has that nucleoid region that contains that ribosomal RNA, that rRNA that I already mentioned. So there's that nucleolus region. And you can see somewhat continuous with this nuclear envelope is this rough endoplasmic reticulum. This kind of enfolded area with all these little dots representing the ribosomes. And then that continues on to a smooth endoplasmic reticulum, smooth ER. So there's both the rough ER, meaning it's rough because it has ribosomes, and the smooth ER is without. And we're going to find out these ribosomes are the actual site of protein production, where we take the information that was originally on the DNA and then transcribed to RNA. We're going to find out it's messenger RNA. That is going to go to encode and make our proteins. And where does that happen? On the ribosomes. All right. So let's look at the ribosomes. They're specialized organelles made up of both RNA, ribosomal RNA, hey, we know that's from this nucleolus, right? And protein. They're found both bound to this rough endoplasmic reticulum as well as just free floating throughout the cell. So just imagine a whole bunch of ribosomes out here. And once again, the ribosomes are the site of protein synthesis. And we'll take a once again, closer look at these a bit later. So what happens is within this nucleus, we have, of course, our DNA. And a segment of DNA will be red. It will be, once again, I'm going to keep on drawing the same stuff out again. It will be transcribed, transcription, all right, into RNA. And that all happens in the nucleus. And then that messenger RNA goes out these nuclear pores to the cytoplasm and will then bind one of these ribosomes, either bound on the rough endoplasmic reticulum or free floating in the cytoplasm. So the messenger RNA will be translated into proteins. And of course, that's in the cytoplasm, either bound to the rough endoplasmic reticulum or free floating. So this is in the cytoplasm, outside the nucleus. But then what happens? We're actually not done there, because after initial translation, other modifications need to be made. And what is this protein going to be used for? So it's making proteins. Well, proteins have lots of different functions. We've mentioned them already. Lots of different structural functions or enzymatic functions. And we're going to talk a lot about enzymes in this module. Either way, they need to be shipped to where they need to go. So they need to be shipped. Packaged and shipped, if you will. Kind of like the post office. Packaged and shipped to their final destination. Well, that's really the role of the endomembrane system. So the endomembrane system is a series of membranes in which molecules are transported in the cell. So right now we're specifically talking about the proteins, but we're going to find out the endomembrane system basically is that post office. It goes both ways. All right? So it goes in and out of the cell, transported throughout the cell. And it constitutes that nuclear envelope, that outer nuclear membrane, right? The endoplasmic reticulum, that ER. And now here are a couple new words: the Golgi apparatus, lysosomes, and vesicles. So let's look at that. All right, so here's a picture straight out of the book, right? And let's start at the bottom, actually. So here's our nucleus. And let's say that we've transcribed some DNA into RNA, messenger RNA, and it comes out, and it's going to connect to some ribosomes, and it's going to be read, and initial proteins are going to be made. But it may not be done there. As you can see, here's that rough endoplasmic reticulum, synthesizes proteins and packages them into vesicles. Oh, well, here that is. See, now what vesicles are simply membrane-bound transports. So we see over here, I'm going to jump to the top for a second, we have an incoming vesicle, and over here we have a secretory vessel, one that's going to be going out. So this one's coming in, this one's going to be heading out. In fact, we can even see that. Over here, we have the plasma membrane, and we can see exocytosis over here, and of course endocytosis over on this side, right? Now, so we have our protein, which may not really be done yet. And this vesicle from the rough ER is going to go over to this Golgi apparatus. Here's our true post office right here. And what the Golgi apparatus does is that it modifies and packages lipids and proteins. So it's not just proteins at this point. Lots of stuff gets handled through here, and basically modifies them. This is where virtually almost all final modifications are going to take place. Others modifications can take place later. We'll find out. But they're getting the protein in our example here ready for its final destination, packages it up, and in this case excretes it out the cell. Now let's look at the other side. Let's go over to the left. Okay, let's take in some molecules through endocytosis, right? We have our incoming vesicle, but maybe we need to break it down. Maybe we need to break this say it's another protein in here, these little balls in here, down to its individual amino acids. Well, the same Golgi apparatus will make something called a lysozyme. And this is almost like your mini digestive system within the cell. So think of it like a little stomach. It contains digestive enzymes that break down cell parts or substances entering vesicles. So what's going to happen is your Golgi apparatus is going to make this lysozyme. It's going to merge, if you will, with that incoming vesicle right here, and notice that that molecule is now digested. It is broken apart. Now let's look. So in a way, you can see this picture is kind of broken into three parts. We've got kind of this protein part right here. We've got this incoming. Okay, let's bring in this big molecule and break it down to its raw components so we can then use it to make our own molecules. But now over here we have a whole little lipid section down in this section in the lower left hand corner. So now we have the smooth endoplasmic reticulum, the one that doesn't have the ribosomes on it, and that actually synthesizes lipids, and it has other functions as well. But for right now, what you need to know is that, hey, here's a lipid inside another vesicle, but this vesicle was created by the smooth endoplasmic reticulum. And same thing happens here. Just as the proteins come from the rough ER and get packaged and goes to the Golgi apparatus, well, here it's coming from the smooth ER in this example and going to the Golgi apparatus for packaging and to its final destination. So, quick summary of the parts of this endomembrane system. Well, we have that rough endoplasmic reticulum, which is studded with ribosomes and used to make proteins. Once again, I hope you have that part down by now. As opposed to the smooth endoplasmic reticulum, it does not have the ribosomes, and that's why, once again, the rough endoplasmic is rough by definition because it has those ribosomes on it. But the smooth endoplasmic reticulum lacks those ribosomes, but they aid in not just lipids, but they also aid in making the carbohydrates that the cell needs. We have that Golgi apparatus, kind of, once again, also more flattened stacks that process, package, and deliver proteins and lipids from that endoplasmic reticulum. And then we have your little cellular stomachs, if you will, those lysozymes, membranous vesicles made by the Golgi that contain digestive enzymes. And in fact, the lysosomes can actually allow the cell to, quote unquote, commit suicide, because the cell can actually rupture all its lysosomes, those digestive enzymes that tends to have a lower pH inside, and it will actually eat apart everything within the cell. So we'll talk more about these concepts a little bit later, but right now the whole concept is, let's look at the main organelles and the processes that the cell uses to make proteins, first and foremost, from DNA to RNA to proteins, but also where does it make its necessary lipids from its raw materials? Where does it make its necessary carbohydrates from its raw materials? And where does it get its raw materials? It'll take it in and digest it from the outside, the extracellular area. And how does everything get transported throughout? And that concept of vesicles. So vesicles is kind of just the go-to term for any small membranous sac used for transport. It could be going in the cell, could be going outside the cell, but the vesicles are what the cell uses to move things around. Here's a little animation from the publisher, specifically on those lysosomes, those membrane-bound sacs that the cell uses for intracellular digestion.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles, such as old mitochondria. This results in the destruction and recycling of these structures.
So obviously your book makes the point that it's even part of the cell's protection where these lysosomes can then break down those foreign materials which can be potentially harmful for the cell. So it creates both a way for the cell to get its raw materials, but also to break down and digest those potential pathogens as well.